Hello, my name is Jaskirat Suri, and this is my Enzyme Lab presentation. Enzymes are proteins that significantly speed up the rate of chemical reactions that take place within cells. They are vital for life and serve a wide range of important functions in the body, such as aiding in digestion and metabolism. Enzymes are highly selective catalysts, meaning that each enzyme only speeds up a specific reaction. The molecules that an enzyme works with are called substrates. The substrates bind to a region on the enzyme called the active site. This binding at the active site lowers the activation energy needed for the reaction, which in turn speeds up the reactions. Trypsin, which is an enzyme that is located in the intestines of humans and is secreted by bacteria, hydrolyzes the peptide bonds located on the carbonyl end of the amino acids lysine and arginine. This is how proteins are digested and broken down into smaller peptide chains and amino acids. For this experiment, we questioned whether trypsin's activity will decrease if we increase salt concentrations. We hypothesized that if the salt concentration increases, then trypsin's activity will decrease due to the fact that the enzyme will be placed in less than optimal conditions. For this experiment, our experimental variables were salt concentrations. We used a variety of concentrations including 0.0 molar, 0.075 molar, 0.3 molar, 0.6 molar, 1.2 molar, and 4.8 molarity. Our control variables were a constant temperature of 25 degrees Celsius and a constant pH of 8. We randomly chose to use 25 degrees Celsius to be one of our constants because we are unaware of the optimal temperatures of trypsin. Our pH was given to us based on our reaction buffers. And our dependent variable was the absorbance at 440.2 nanometers. When conducting the experiment, we first added 450 microliters of the reaction buffer, 500 microliters of azocasein, and 50 microliters of trypsin to a clean centrifuge tube. We then let it incubate for 10 minutes. Azocasein is a milk protein with sulfanilamide attached randomly. The purpose of adding azocasein is to allow absorbance at the 440 nanometers. After 10 minutes of incubation, we added 500 microliters of a 10% TCA solution. The purpose of the TCA solution is to stop the reaction and precipitate any undigested substrates. We then centrifuged the test tube at a maximum speed in the clinical centrifuge for 2 minutes. The purpose of centrifuging the test tube was to remove any undigested substrate. We then transferred the supernatant into a clean cuvette. We blanked the spectrophotometer at 440 nanometers using a cuvette with distilled water. We then recorded our results and repeated this process for our 3 trials and our negative control. The negative control was prepared very similar to our test tubes. Instead of adding trypsin to the negative control, we added distilled water to ensure that trypsin was the responsible agent for the reaction. We used a total of 3 replicates, not including our negative control, in order to reduce variability and assure validity. Activity decreased as the salt concentration increased from 0 molar to 4.8 molar. Activity at 4.8 molar was significantly different from that at 0.075 molar, but at the rest of the salt concentrations were not significantly different from the 0.075. Variability among all the trials was very small, and they all showed the same conclusion, which was that the higher the salt concentration, the lower the enzyme activity. We placed our data in a graph based on the averages, and it showed the decrease in activity as the salt concentration increased. So this is a copy of our graph. It basically shows the comparison in the enzyme activity between the sodium concentration and the absorbance levels. So the graph basically shows the decrease from the 0.0 molar. Our graph, you can't really tell the difference between the 0.0 molar, the 0.075 molar, and the 0.3 molar, but the 0.075 was a little bit higher than the 0.0 and the 0.3 molar, but overall you can see the trend as you increase the sodium concentration, the absorbance decreases, which means that the enzyme activity also decreases. So Originally, we had hypothesized that increasing the sodium content of the solutions made in the trypsin enzyme assay would decrease trypsin activity. All the experiments were performed at room temperature and a neutral pH of 8. The hypothesis was supported based on the data collected from the experiment. The concentrations of the sodium used were 0.0 molar, 0.075 molar, 0.3 molar, 0.6 molar, 1.2 molar, and 4.8 molar. Trypsin activity peaked at 0.075 molar of the sodium, resulting in a reading of 0.173 at trial 3, before beginning a downward trend with each subsequently larger sodium concentration being tested. The lowest reading was 0.046 during trial 1 using the 4.8 molar solution. We can conclude that this experiment of increasing salinity is not conducive to the growth of bacteria as it decreases enzyme activity. Referencing Group D, who tested enzyme activity with varying temperatures, it is clear that the temperature had a greater effect on enzyme activity than the salinity, with the results of 50 degrees Celsius peaking at a measurement of 0.643 during their first trial. The source of variability may be due to incorrect measurements of the reaction buffer, azocasein, or the trypsin. Variability may also be due to the fact that our absorbance level was at 440.2 nanometers instead of exactly at 440 nanometers. We can improve further experiments by increasing the number of replicants as well as making sure that our measurements are as accurate as we can make them.